Good evening, everyone. I welcome you all to this session today. Today, we are conducting a session of PRAGNAVARIDHI lecture series. We have Vaidya Krishna Kumar sir. Sir will be teaching about a Haemopoietic system and immunity, mainly exam oriented session. Panchajanya, the divine conch of Bhagavan Sri Krishna, which was blown during Kurukshetra, indicating the start of Dharma Yuddha that followed. There are all the efforts were done for the establishment of Dharma. Dharma was kept over personal relations and motives. Keeping the same motive in mind, our organization will work towards establishing the importance and necessity of following and imbibing the culture, accepting our roots and making people realize the true essence of Ayurveda, not only as a treatment modality but also a way of life. All these while working in the path of Dharma. Hence the motto Shreyam Bharatiyam Arogyam Ayurvediyam. The founders of this organization started this because we felt the need to spread the knowledge of Bharatiya culture, Ayurveda, and its values to all strata of people in every corner of the world. Also, the founders being Ayurveda aspirants know the inside out of today's medical field, hence, they aim to make people aware of this and also try their best to solve this on the go. In order to follow all these to be achieved, we have set up various wings with working principles and objectives that help achieve the goals faster and easier in the coming years. They are Team Swami Vivekananda, Team Ashwins, Team Veda Vyasa, Team Sanjaya and Nalanda. Coming back to the members involved in our organization, we have doctors, healthcare workers, teachers, students, and people from almost every single occupation available aiming at working together and bringing forward the true essence of Bharat and her traditions. So, all those who fit under these categories are welcome with honor and privilege. In this today's session, we have Vaidya Krishna Kumar sir, currently working as professor and HOD, Department of Kriya Shareera, Enopia Ayurvedic Medical College, Mangalore. Sir has completed both UG and PG from Government Ayurveda College, Kannur, Kerala, under Kerala University of Health Science. Sir has participated, presented, and chaired various national and international seminars. We are glad to have you in this session today, sir. We'll start the session now. Over to you, sir.
Thank you. Thank you for a warm welcome. Hi, all. A very good evening to all. We are already late by 15 minutes. So, first of all, today is this June 21st, is the International Yoga Day. So, Yoga Day wishes to all. I'm very happy to be here as a part of this knowledge sharing lecture series. Pragnavaridhi is conducted by the Panchajanya organization. Actually, this Panchajanya organization, after I heard about the Panchajanya organization, it connects all the people. So the word meaning of yoga is that which union, that which unite all. So here on the online platform, you connect all the people through the knowledge. So it is a very good program, and I am very thankful to the organizers for inviting me as a speaker. Today, the topic given to me is hemopoietic system and immunity. It is only an exam oriented class because we already covered in all colleges. The coming month, the next month will be the exam. So we are refreshing the already studied knowledge, and so it's an exam orientation. So let's move on to the topics. Next slide. Previous slide. Back. Yeah. So come to the mark allotment. This only on exam point of view. The topic hemopoietic system and immunity comes under the paper 2. Kriya Shareera is having two paper 1 and 2. And in paper 2, the part B is the modern part and it for 40 marks. And part 1 is the Ayurveda portions, that is the Dhatu and all that which comes under the part A, and it is for the 60 marks. So 60 is to 40, that is according to the new NCISM. And among these, the part B 40 marks, among these, the 80 marks from blood immunity and CVS among the 40 marks, 80 marks. About to 50% is from this blood or hemopoietic system, immunity and cardiovascular system. So that's the importance of this system. And from this area, we will get the questions like MCQ, short answer and long answer questions. MCQ and short answer from the immunity. In the immunity, MCQ and short answer will be there, but from the hemopoietic system and CVS, both all these MCQ, short answer and long answer questions will come. And the total allotted time given to us is the 5 lecture hours and 2 non-lecture hours. So the Ayurveda 60%, modern 40%. So the first part, the blood and hemopoietic system and immunity is the today's topic. So next, the syllabus includes main topics covered. We have to cover is blood and properties, composition and functions of blood, then plasma proteins and functions, erythropoiesis and the stages and factors controlling erythropoiesis, and structure, types and functions of hemoglobin, anemia and the types of anemia, and hemostasis and blood coagulation, immunity and types of immunity. These are the main topics, and the question may ask what is the blood and properties, composition and functions of blood for 5 marks. Like that, the question may come. Erythropoiesis, stages and factors controlling erythropoiesis. That question may come for 10 marks. This type of questions may ask from this hemopoietic system. Next slide, please. So coming to the first topic, blood and what is blood and the properties of blood. That we already know what is blood, and blood is the only fluid connective tissue and that which flows through the vascular system, and it is that which carries respiratory gases, oxygen and carbon dioxide. So the color, what are the properties of blood? The color of the blood, that is red in color, and the volume is 5 liter. The color may be changes according to the arterial and venous, the scarlet red and purple red according to the text. And then volume is 5 liter, and the reaction is slightly alkaline with the pH 7.4, and specific gravity 1.05 to 1.061, and the viscosity is 5 times more viscous than water because the presence of plasma protein and the blood vessels. So the properties, what is blood? Properties of blood, that is important. Next slide. And the composition of blood, it is also important. Composition of blood, that which contains the plasma and the formed element. Plasma is the fluid portion, that is 55%, and formed element forms the 45%. Formed elements are the blood cells, that the blood corpuscles are the red blood corpuscles and white blood corpuscles and the platelets. And among these white blood cells, granulocytes and agranulocytes are there. That is the granulocytes, the granules are present, neutrophil, eosinophil, basophil, and the granulocytes are the lymphocyte and monocyte. And next slide. And the plasma, and the plasma is the fluid part of the blood, and it is 55%, and among this plasma constitute water and the solids. Solids contain the protein and others, and the water composition contains about 90 to 92% is water, and remains 8 to 10% is the solids, and the solids includes the plasma proteins. Up to 78% is the plasma proteins that we know: albumin, globulin and fibrinogen. These are the plasma proteins, and others not slide, previous slide. So among these, the solids, these are the plasma proteins are there. Then amino acids, essential amino acids, non-essential amino acids are there. Sugar, glucose is there. Fatty acids, cholesterol, all others are there. And then nitrogenous waste product, the urea, uric acid, ammonia, xanthine, hypoxanthine, all is present in the blood, and different enzymes, serum amylase, serum amylase, alkaline phosphatase, acid phosphatase, and then carbonic anhydrase. Different enzymes are present. So plasma composition is important, water and solids among organic and inorganic. Inorganic comes under the sodium, potassium, calcium, carbonate, and then sulfate, chloride, etc. So this question may ask for blood, composition of blood and functions. Next is the functions of blood. Next slide. please. Functions of blood. Functions of blood that we know, the first one is the nutritive functions. After the digestion, all the nutrients are absorbed from the intestine to the blood, and then this blood that which carries the nutrients and oxygen to all the tissues and the nourishment occurs, that we know. So the nutritive function. Respiratory functions, we know respiratory function, how the oxygen is carried from the lungs to the tissues, and then the metabolic waste product, that is CO2 and other waste material, are carried from the tissues to the exterior. So respiratory function is also main blood function. Excretory functions, after the metabolic waste products all are reach into the excretory organs like liver, kidney, skin, etc. So blood is having excretory function also. So transport of hormones and enzymes. We know that the blood, the hormones are the chemical messengers directly enter into the blood, and that which transports through the blood. So blood act as a transporting mechanism. And regulation of water balance, ECF volume, that we know the water balance is also controlled by the blood, and acid base balance, hemoglobin is present and the plasma protein is present, so it is act as buffers, so it maintains the acid base balance. And regulation of body temperature is also maintained by the blood. And storage function, it's an important storage function. Plasma proteins and then water is there, so whenever the dehydration is there, the water is drained from this blood. And also defensive function is the defensive function. Main important function of blood is a defensive function by the lymphocyte, neutrophils, macrophages are involved in the defensive function. T lymphocytes are in the cellular immunity, B lymphocytes are in the humoral immunity, that I will explain later. So these are the functions. So these are first one question came from this part. Next is the plasma proteins, and next slide. Next question may ask is the what are plasma proteins and properties and functions of plasma protein. For 5 marks may ask. So the plasma proteins are the important plasma proteins are serum albumin, serum globulin, and fibrinogen. Why we are calling it fibrinogen? Serum albumin and globulin, these are present in the serum, but fibrinogen is not present in the serum because the fibrinogen is converted into fibrin. So it is called as fibrinogen only, not serum fibrinogen. So these are the main plasma proteins, and the normal value of plasma proteins are 4.7 gram per deciliter serum albumin, serum globulin is 2.3 gram per deciliter, and fibrinogen is 0.3 gram per deciliter. This is important. And total protein 7.3, that is the average of 6.4 to 8.3. And there is an important point is serum albumin globulin ratio is the A by G ratio. That is normally it is 2:1, and in pathological condition, liver diseases and other pathological conditions, this A by G ratio reverses. So it's indicator of liver disorders. That is important. Next, the properties of plasma proteins. Plasma proteins molecular weight, that we already saw that in the previous slide. Just previous slide. Just go to the previous slide. The molecular weight we can see here. Serum albumin is having less 69,000, and the highest molecular weight is having fibrinogen, that is 4 lakh. So this is the molecular weight of the plasma proteins. Next is the oncotic pressure. Oncotic pressure is the osmotic pressure exerted by the plasma proteins in the blood, that is 25 mmHg, and mainly the pressure exerted by the plasma protein albumin. The main contribution of oncotic pressure is by the albumin, and the specific gravity is 1.026, and it is having buffer action. Plasma proteins is having buffer action, is having albumin is having the buffer action. So these are the properties of plasma proteins. Next slide. And functions of plasma proteins. Its role in coagulation of blood, that we will discuss later, that we know coagulation is the formation of clot. Coagulation, the fluid blood is converted into gel. So fibrinogen is converted. Fibrinogen is the protein, here plasma protein. So role is in coagulation is important. Role in defense mechanism of the body. We know that the defense mechanism, immunoglobulins are there. Antibodies are immunoglobulins or gamma globulins. So in defense mechanism is also important. In role in transport mechanism, This transport mechanism, we know that the hormones and enzymes are transported by these plasma proteins. Albumin, which helps the transport of bilirubin, we know, conjugated bilirubin. This along with the free bilirubin bind with the plasma protein albumin and reach into the liver. So it acts as a transport mechanism, role in transport mechanism. And maintenance of oncotic pressure that we already studied. Oncotic pressure is the osmotic pressure by the plasma proteins. And regulation of acid-base balance, and this acid-base balance is regulated by the plasma proteins, albumin mainly having buffer action. Role in viscosity. Viscosity is by these plasma proteins. Normal blood is more viscous. Blood is more viscosity, having more viscosity because of the plasma proteins. The concentration increases. And role in ESR, erythrocyte sedimentation rate. Erythrocyte sedimentation rate, albumin, that which prevent the rollers formation, but this fibrinogen and the plasma albumin globulin, that which helps for the rollers formation and that which increases the ESR that we know. And role in suspension stability of RBC. Normally, in the flowing blood, the blood cells are suspended because of the help of these uh, plasma proteins like globulin and fibrinogen. That is why they are not stick onto the blood vessel wall. Suspension stability is an important function. And role as reserve protein in starvation and dehydration. In starvation, we are not taking food at that time. We are using that the reserve protein as these plasma proteins. And role in production of triphone bodies that which nourish the cells in the tissue culture. So these are the main functions of plasma proteins. Next slide. So two questions over. And third question, erythropoiesis and stages and the factors controlling erythropoiesis. Very, very important area. This is a must know area, and for 10 marks maybe ask. So what is erythropoiesis? We know the process of origin, development and maturation of RBC is called as erythropoiesis. Hemopoiesis means the process of origin, development and maturation of blood cells. Then it is known as hemopoiesis. And the site of erythropoiesis, it is different in different level. In fetal life, it is having three stages. That is, first one is the mesoblastic stage. Mesoblastic stage in the first 1 or 2 intrauterine life, the here the RBCs are produced from the mesenchyme of the yolk sac. And the third month onwards, the hepatic stage, the RBC is produced from the liver, and then myeloid stage, the last trimester, the myeloid stage. Here, the bone marrow, from the bone marrow, the RBCs are produced. And in newborn babies, children, and adults, the RBCs are produced from the red bone marrow. But up to the age of 20, the red bone marrow of the for both the flat bones and long bones, red bone marrow that which produce the RBC. But after the age of 20, the red bone marrow in the only in the end of long bones, and the flat bones or membranous bones like ribs, vertebra, and scapula and skull. Only in this part, the red bone marrow that which produce the RBC. So the site of production of RBC is different in different ages. Next. Next slide. So during this erythropoiesis, there are different changes occurring to the cells, and the major changes are reduction in the size of the cell. So that is from the diameter of 25 to 7.4 mu. That is the first one. The important point is the reduction in the size of the RBC. Second one is the disappearance of nucleoli and nucleus, and then appearance of HB. And the fourth one is change in the staining properties of the cytoplasm. These are the four main changes that which occurring at the time of erythropoiesis. Next slide, please. So this is the picture that will show the erythropoiesis changes, biotic changes. We know that here we can see the pluripotent hemopoietic stem cells. That is first, the hemopoietic stem cells are the chief primitive cells that which produce specialized cells. Here the pluripotent hemopoietic stem cells that is inside the red bone marrow. And these hematopoietic stem cells that which produce two types of colony forming blastocyte and lymphoid stem cells, two types of cells. And this colony forming blastocyte is again different colony forming units are formed. That is colony forming unit E, colony forming unit M, and colony forming unit GM, that is granulocyte monocyte. Then this lymphoid stem cell, that is lymphocyte that which give rise to the lymphoblast and the lymphocyte, and all other colony forming unit, that blastocyte which produce the colony forming unit erythrocytes, colony forming unit megakaryocytes, and that which produce the platelets. Like that we can understand from this picture. So this is the changes that which occurring during the erythropoiesis. Next slide. So stages of erythropoiesis is also important. In the textbook we can see that the diagram is a we can at the time of exam you can write the important points. So here the stages of erythropoiesis are mainly 6 stages. We can see the pro-erythroblast, early normoblast, intermediate normoblast, late normoblast, reticulocyte, and matured RBC. These are the 6 stages of erythropoiesis. And we can see that the changes, first one, the changes in diameter, which are the changes. First, we noticed that the size reduction. First one, the pro-erythroblast is the first stage. From the colony forming unit, the pro-erythroblastic stage, or it is known as megaloblast, because it is large in size. It is having 20 mu in diameter, and has 2 or more nucleoli and a chromatin network. A nucleus is present, and then staining property is basophilic, and synthesis, and there is no hemoglobin present in the pro-erythroblast. Only the synthesis of hemoglobin starts, but not present in the pro-erythroblast. That is why this staining is basophilic. So these are the important points that which in pro-erythroblast. So next stage, the multiplication occurs, and the early normoblast is the next stage. And in this, the diameter reduced to 15 mu, and here no nucleoli. The disappearance of the nucleoli and the chromatin network, that which caused the condensation, and the dense chromatin network and nucleoli disappearance is the important point in that stage. And third stage is the intermediate stage or intermediate normoblast. Here again the diameter reduced, further condensation of chromatin network occurs, and here important thing is hemoglobin starts appearing. It is the stage during which the hemoglobin starts appearing. We can see the hemoglobin. It's an important point. So in the late normoblast, again the diameter reduced 8 to 10, and it is small with very much condensed chromatin. And here another one is ink spot nucleus. That the word you write it as ink spot nucleus. That is an important. And this staining property become acidophilic because of the presence of hemoglobin. And nucleus disappears by pycnosis. It's an important part. Pycnosis, disintegration of the nucleus. So late normoblast. And then reticulocyte, the size diameter becomes 7 to 7.5 mu, and the nucleus is absent. That is the important point. Reticulocyte nucleus is absent, and the basophilic, the staining property. Reticulum is formed. That is why it is this stage is known as reticulocyte. Reticulum is formed by the disintegration of the cell organelles. And the cell enters capillary from site of production. That is the reticulocyte. So the time taken from the pro-erythroblast to the reticulocyte is 5 days, and from the reticulocyte to mature RBC it will take 2 days. That is an important point. So mature RBC, the final mature RBC is having 7.2 mu diameter, and nucleus non-nucleated biconcave RBC formation, and it is acidophilic in nature, and cell attains biconcavity. These are the different stages. So very, very, very important: pro-erythroblast, early normoblast, intermediate normoblast, late normoblast, reticulocyte, and matured RBC. So these are the important stages of erythropoiesis. So first, what is erythropoiesis? Different stages. And then last part of the question, factors for erythropoiesis. The factors for erythropoiesis, 3 factors are the stimulating factors, maturation factors, and factors necessary for HB formation. These are the three important factors. Next slide. Stimulating factors are the hypoxia, erythropoietin, thyroxine, sex hormones, hemopiotic growth factors, and vitamins. These are the stimulating factors. During hypoxia, the erythropoietin secretion increases, and that which produces more RBCs. And thyroxine also stimulate the production of RBC, sex hormones, and hemopiotic growth factors like interleukin, which helps for the which act as stimulating factors. And vitamins B3, B2, B3, B6, B12, etc. are also stimulating factors. And maturation factors are the vitamin B12 and intrinsic factor of calcium and folic acid. And we know vitamin B12 is known as the extrinsic factor of calcium. That is, we are getting from the folic acid, and intrinsic factor of calcium is so the vitamin B12, intrinsic factor of calcium, and folic acid all are. Am I audible?
Yes, sir, you're audible, sir.
Okay, so the vitamin B12, intrinsic factor of calcium, and folic acids all are helps for the synthesis of DNA and the maturation of RBC. So in the absence of this vitamin B12 and folic acid, that which leads to a large sized RBC, that is folic acid deficiency leads to megaloblastic anemia. We already saw that megaloblast is the pro-erythroblast, that the size of the RBC is large. And in the also vitamin B12 deficiency leads to pernicious anemia. These are two important points. And the factors necessary for HB formation, here the first class proteins and amino acid, that which helps for the production of hemoglobin. Then the globin part is the protein. So these proteins are important. Iron helps for the heme part of the hemoglobin. Iron is needed. And the copper, that which helps for the absorption of iron from the intestine. And cobalt and nickel are also important, and vitamins are important, necessary for the hemoglobin formation. So these are the important things that cover in the maturation and the process of erythropoiesis. Factors controlling erythropoiesis. So then next question comes, maybe come from the hemoglobin. What is hemoglobin? Structure and functions of hemoglobin. So hemoglobin, we already know that the hemoglobin is the iron-containing coloring pigment of the RBC, and it is a chromoprotein forming 95% of the dry weight of RBC. And the function is to carry the respiratory gases like oxygen and carbon dioxide, and it act as a buffer. Hemoglobin act as a buffer, and molecular weight is 68,000. Important thing you should write at the time of exams. Only important points I am covering. And the structure is, it's a conjugated protein combined with the iron containing pigment. The iron containing pigment is the heme part, that is the porphyrin is the pigment part of the heme, and it is normally in ferrous form. And protein part is the globin, and it is formed by globin contains 4 polypeptide chains like 2 alpha and 2 beta chains. And the porphyrin ring is formed by the pyrrole ring. 4 pyrrole rings are attached by methane bridges. We can see the picture, the structure of hemoglobin. Next slide. And next is the types of hemoglobin. We already know hemoglobin that carries oxygen and carbon dioxide, respiratory gases. Two types of normally physiologically, two types of HBs are present, that is adult hemoglobin and fetal HB, HbA and HbF. In adult hemoglobin, structural and functional differences are there in both. And the function structural difference is that in adult hemoglobin, the polypeptide chains, two alpha chains and two beta chains are present. And in fetal hemoglobin, two alpha chains and two gamma chains are present. That's the structural difference. And the functional difference is that the fetal hemoglobin is having more affinity for oxygen than the adult hemoglobin. This is a functional difference. So these are the two types of hemoglobin. Next is the abnormal hemoglobin. So abnormal hemoglobin may be produced by pathological conditions or mutations that which happens to the hemoglobin. So abnormal hemoglobin in hemoglobinopathies and abnormal hemoglobin in thalassemia and related disorders we can see. So in hemoglobin, abnormal hemoglobin in hemoglobinopathies, like example hemoglobin S. In sickle cell anemia, normally alpha chains and beta chains are there, and in the abnormal beta chains are present in this hemoglobin S. Instead, here the substitution of amino acid valine for glutamic acid at the sixth position of beta chains. That's the important thing. So here hemoglobin S. It's an abnormal HB in hemoglobinopathies that we can see in different hemoglobin C, E, H, and M. We can see. And abnormal HB in thalassemia and related disorders. In thalassemia related disorders, here the polypeptide chain is having sometimes both the alpha thalassemia and beta thalassemia occurs. Alpha thalassemia means the alpha chain, polypeptide chain is sometimes decrease in number, or sometimes absent, or sometimes abnormal polypeptide chains are there. That is the thalassemia. Alpha thalassemia and beta thalassemia are there. These are all abnormal hemoglobin. Next slide. Next, hemoglobin derivatives. These are the hemoglobin derivatives are the carboxyhemoglobin, methemoglobin, and subhemoglobin. We know normal function of hemoglobin is carry oxygen. So his HB derivatives are the abnormal hemoglobin that cannot carry the oxygen. So it can lead to the hypoxia in the tissues, because carboxyhemoglobin formed by the combination of carbon monoxide. Carbon monoxide bind with the hemoglobin to form the carboxyhemoglobin, and this carbon dioxide is carbon monoxide is having, not carbon dioxide. Carbon monoxide is the carboxyhemoglobin, and this carbon monoxide is having 200 times more affinity to oxygen hemoglobin than oxygen. 200 times more affinity to hemoglobin. So carbon monoxide poisoning, that which leads to dangerous conditions. Methemoglobin, here the ferrous form is oxidized into ferric form. The stable ion is the ferric form, so it cannot carry the oxygen. And sulfhemoglobin is formed by the combined with the iron sulfide here. So these are the HB derivatives. And the functions of hemoglobin is transport of respiratory gases, oxygen from lungs to tissues, and CO2 from tissues to lungs. And is also having buffer action, the rolling acid-base balance. So important question: hemoglobin, types of hemoglobin, and abnormal derivatives of hemoglobin, and the functions of hemoglobin also important. Next, another important question: anemia and types. When I am taking classes, always I am telling like that. Anemia and types of anemia is very, very important. So we all know that anemia is a blood disorder characterized by the reduction in the RBC count, hemoglobin count, and then packed cell volume, PCV. Because we know that packed cell volume, PCV, is the percentage. This RBC expressed in percentage, red blood cells expressed in percentage. It is also known as hematocrit value. So the classification, broad classification of anemia is the morphological and etiological classification. The morphological classification depends on the size and the color of the RBC. Size determined by the mean corpuscular volume, MCV, blood indices. This comes under the blood indices, and it is MCV, mean corpuscular volume, and the color is indicated determined by the MCHC, that is mean corpuscular hemoglobin concentration, the presence of hemoglobin in the blood. So morphological classification. Next slide. So morphological classification again divided into four types: normocytic, normochromic, macrocytic, normochromic, macrocytic, hypochromic, and microcytic, hypochromic. The word itself is having the meaning. Normocytic, normochromic means the size is normal and the color is normal. Normocytic and normochromic. Here the size MCV is normal and the MCHC is normal. Macrocytic, normochromic means large size but the color is normal. Macrocytic hypochromic means large size but the color less, hemoglobin less. And microcytic hypochromic means both the color and the size decreases. So in different types of anemia, we can see this different types of this presentation. Next slide. Next is the etiological classification. Etiological classification, mainly the five types of anemia are explained. Etiological, the causes behind the anemia is divided into types of causes. First one, the hemorrhagic anemia, then hemolytic anemia, nutritional deficiency anemia, aplastic anemia, and anemia due to chronic diseases. So coming to the first one, hemorrhagic anemia, the word itself is having the meaning hemorrhagic, excess loss of blood by internal or external bleeding. And it may be occur acute bleeding and chronic bleeding. Acute bleeding occurs in the conditions like emergency accidents. So a large loss of blood occurs. And in this condition, more amount of blood, acute loss of blood from the body. So here only the reduction of RBC occurs, and the this blood indices morphological classification comes under in acute hemorrhage, normocytic, normochromic anemia, because the size and color of the RBC is normal, but if the RBC count only decreases. That is a type of anemia in hemolytic acute hemorrhage. But in chronic cases, the size and means due to the loss of blood due to peptic ulcer and then pile mass, bleeding pile mass, and also the menorrhagia, increased blood loss, that which continuous loss of blood for a long duration, that which leads to decreased size and iron content is less. So microcytic and hypochromic anemia may happen. That is the hemorrhagic anemia. Second type is the hemolytic anemia. Hemolytic anemia is occur by two types: extrinsic hemolytic and intrinsic. Extrinsic means the destruction of RBC occur due to the causes outside the blood, and intrinsic inside the hereditary causes, inside the causes, blood itself, that which causes the destruction. So the extrinsic hemolytic anemia occur due to diseases like liver failure, renal disorder, hypersplenism, and then burns, infections like hepatitis, malaria, etc. And the intrinsic hemolytic anemia, hereditary disorders like hemoglobin S, we already see that sickle cell anemia in this condition, thalassemia related disorders, hemolytic anemia occurs. And in the hemolytic anemia also, normocytic and normochromic anemia may be there. And nutritional deficiency anemia, it occurs due to the iron deficiency. The most common type of anemia is the iron deficiency anemia, and protein deficiency is there, then it will lead to anemia. Or vitamin B12 deficiency, that which leads to pernicious anemia, and folic acid deficiency leads to megaloblastic anemia, that already explained. And in iron deficiency anemia, the morphology is microcytic and hypochromic anemia. And protein deficiency, macrocytic and hypochromic anemia. So different types according to the cause. And aplastic anemia is the bone marrow disorder due to the continuous or prolonged exposure to the X-rays and all. So it will lead to aplastic anemia. And in aplastic anemia, the morphology is normocytic and normochromic. And the last one is the anemia due to chronic diseases. And here, bone marrow, here the non-infectious inflammatory disorders like rheumatoid arthritis, in all this case, anemia occurs. And then here, this is the etiological classifications. Next slide. Next is the hemostasis. Hemostasis, the arrest or stoppage of bleeding is the hemostasis. It occurs in three stages. That is, the three stages are vasoconstriction, platelet plug formation, and coagulation of blood. Next slide. This may also ask for the 5 marks hemostasis. Three steps. So when an injury to the blood vessel occurs, that which cause the damage of the endothelium and exposure of the collagen fibers, and the collagen exposure of the collagen, and then the adherence of platelets to collagen, that which activate the platelets. So the platelets are activated by the exposure of collagen, and then that the activated platelets that which secrete the serotonin, and this serotonin that which causes vasoconstriction. And that vasoconstriction is purely local phenomenon, and that which decrease the blood flow. First stage is the vasoconstriction, and it is a local phenomenon, and the constriction of arteries and arterioles occurs. That is vasoconstriction stage is the stage 1. Next is the activated platelets. More and more platelets are adhered to the collagen, and then secretion of ADP and thromboxane A2 are produced from the activated platelets, and platelet activating factors are there, and more aggregation of platelets occurs, and that which leads to platelet plug formation, and that is the second stage, platelet plug formation. And the third stage is the formation of prothrombin activator and the platelet blood clotting mechanism. So these are the three stages. So hemostasis is the stoppage or arrest of bleeding, that which is having three stages: stage of vasoconstriction, platelet plug formation, and blood coagulation. These are the 5 marks may ask. So you can write like this. This diagram you can draw. This chart you can draw. So next slide. And then next is the coagulation mechanism. So coagulation, blood coagulation is important thing. That means the blood is having the fluid connective tissue, that which loses its fluidity and become gel-like mass. Already I told, it becomes gel-like mass because the fibrinogen is converted into fibrin. So different the enzyme factors that which helps for the clotting mechanism, and all these factors are called clotting factors, and these clotting factors are present in the form of clot enzymes. These are proteins present in the form of enzymes, and this mechanism is known as cascade mechanism, because one enzyme, these enzymes are in the inactive state, that is pro enzyme, and then once it activated, then it will activate the next step and the next step till the end of the functions, so end of the reaction. So it is known as cascade mechanism. So the blood coagulation mechanism is a cascade mechanism. So here first two stages over, then blood coagulation. For this we need the formation of prothrombin. First one is the formation of prothrombin activator. So the formation of prothrombin activator occurs by two mechanisms: intrinsic pathway and extrinsic pathway. In intrinsic pathway, the cause is inside the blood itself, that is the blood platelets. And the extrinsic pathway, the tissue trauma occurs, and the tissue thromboplastin is responsible for the production of prothrombin activator. So in the first stage is a intrinsic pathway. Here, first one, after the damage, endothelial damage and collagen exposure is there, then this factor 12 will contact with the collagen, and then activated factor 12 formation occurs with the help of presence of kallikrein and a high molecular weight kinogen. And this activated factor 12, that which form activate the factor 11 to activated factor 11 in presence of high molecular weight kinogen, and this activated factor 11 convert the factor 9 into activated factor 9 in presence of calcium. It's a cascade of reactions, and at last the activated factor 9 convert the factor 10 into activated factor 10 in presence of 8th factor and calcium. And this activated factor 10 combined with the phospholipids, this is the when the endothelial collagen exposure and the platelets, activation of platelet activates, and this produce the phospholipids, and these phospholipids activated factor 10, and in presence of 5th factor, and in presence of calcium, that which form the prothrombin activator. This is the intrinsic mechanism. But in extrinsic pathway, here the tissue thromboplastin is involved, and here the tissue thromboplastin is having glycoprotein and phospholipids, and this glycoprotein and phospholipids combined with the uh, activates the factor 10 into activated factor 10, and this activated factor 10 in presence of this glycoprotein and calcium and fifth factor, that which form the prothrombin activator. In presence, stable factor is needed. Stable factor, the seventh factor is the stable factor. So all the factors involved in the blood clotting mechanism, there are 13 factors are involved in the mechanism. First one is the fibrinogen, second prothrombin, thromboplastin, calcium, labile factor, and factor not poor, stable factor, and the hemophilic factor, Christmas factor, Schwartz growth factor, plasma thromboplastin antecedent, then Hageman factor, fibril stabilization. These type different 13 factors are there. So here in extensive pathway, the stable factor is involved. Seventh factor, along with the glycoprotein, phospholipids, and factor 10, activated factor 10, and then fifth factor on calcium, prothrombin activator formation, and then this prothrombin activator formation, and this convert in the prothrombin to thrombin formation. Second stage is the conversion of prothrombin to thrombin, and it is the main important thing in the blood coagulation mechanism, formation of thrombin. And this formed thrombin, there is a mechanism, is the positive feedback mechanism. This formed thrombin, which will activate the formation of fifth factor, that which increase the formation of prothrombin activity. This is a positive feedback mechanism is there. And this thrombin, that which convert the fibrinogen into fibrin. This fibrin is the active fibrin monomer. Inactive fibrinogen into active fibrin monomer, and the polymerization of the fibrin monomer, first the loose strands of fibrin, and then with the help of factor 13, name is fibrin stabilizing factor, and calcium, in presence of calcium, tight fibrin blood clot formation or the coagulation mechanism occurs like this. So this is an important area. Also ask for 5 marks or 10 marks, hemostasis and blood coagulation mechanism. So you go through all this and you can draw the picture like this. Next slide. Immunity and types of immunity. And from this area, only MCQ and short answer questions may come. Not long answer, not for the 10 mark question, MCQ and 5 mark question, 5 marks. Then here immunity and types. The question may ask for like what is immunity and types of immunity, and explain in detail about cell mediated immunity or humoral immunity, like that the question may ask. We can see in the previous question paper. So what is immunity? Immunity is the capacity of the body to resist the invading microorganisms. So the types of immunity, the broad classification of immunity is the innate immunity and the acquired immunity. Then what is innate immunity? Innate immunity is the inborn ability of the body to resist the invading microorganism. There are different, if any way this enter into the body, this innate immunity will block the entry of that microorganism and prevent the formation of diseases. So in different systems, this innate immunity works. And in GI system, we know that in GI system, GI enzymes are there, that which prevent the entry of microbes through the food. And also the acids present in the stomach, hydrochloric acid, that which prevent the entry of microbes through this food. And in saliva, lysozyme is present. It is also works against the microorganism. So GI system, it is works like that. Innate immunity is working like that. And in respiratory system, defensins and cathelicidins present in the epithelial cells in the respiratory tract, that which is having the action of antimicrobial property. And also the neutrophil and macrophages in the lungs, that which are also act as the innate immunity. So different urogenital system is having acidic urine, that which prevent. So different system innate immunities we are having. So next slide. So we can see here innate immunity and naturally occur, we can see. And the next one is the acquired immunity. Acquired immunity is the important one, that which acquired immunity, the word itself is having acquired after exposing to any microorganism. We are acquiring the immunity after specific microorganisms like viruses, bacteria, fungi, and toxic substances. Here, the innate immunity is non-specific in nature, but the acquired immunity is specific in nature, that which specific to the organisms. Like in acquired immunity is specific in nature. That is the important difference. So acquired immunity is again divided into two: cellular immunity and humoral immunity. So while answering the question, you should write that the definition of immunity and the basic classification of immunity. That is first one, innate immunity and acquired immunity. And then acquired immunity is again divided into two. You can draw this diagram, flowchart, and the acquired immunity is again cellular immunity and humoral immunity. First one is the cellular immunity. The word itself is having the cellular immunity. Cellular immunity is defined as the immunity is mediated through the cellular response, cell mediated response, that we can say. The different types of cells are involving in the cellular immunity. It is also known as cell mediated immunity. And different types of cells like T lymphocytes. T lymphocytes are involving, and then macrophages are there, natural killer cells are there. So different types of cells are involving in this immunity. So it is known as cellular immunity, and it is more specific against the viruses and fungi. So it is specific in nature. So before that, how it will occur, the mechanism of cellular immunity. So before that, lymphocytes are of different types. T lymphocytes, B lymphocytes are the lymphocyte. T lymphocytes processing the thymus, and B lymphocytes are processing in the birds of cloac of birds, fabricaceous birds of fabricaceous. That is why it is known as B lymphocytes. So T lymphocytes are helps for the cellular immunity. So how the mechanism of cellular immunity that we can look. First, an antigen is needed when the antigen presenting cells are there. So antigens are the agents that which produce the immune reactions in the body. Antigens are the proteins. So when any type of microorganism that which enter into the body, so the digestion occurs and the antigenic peptides are formed. These are protein, and these antigens are presenting to the T cells, T lymphocytes. So T lymphocytes are of four types mainly that you should write. Four types of T lymphocytes are: first one is helper T cells or T4 cells, helper T cells, or inducer cells. Different names are there: helper T cells, T4 cells, and CD4 cells, helper T cells, and then killer T cells, cytotoxic T cells or killer T cells or CD8 cells, and then suppressor T cells, and or regulatory T cells and memory T cells. These are the different types of T cells. So how the activation of helper T cells occurs when an antigen enter into the body, and then this antigen presenting cells, different types of antigen presenting cells are present in the body, like macrophages are there, dendritic cells are there. So this antigen presenting cells that which present the antigen to the helper T cells. So the specific helper T cells are there. So the changes occurring when the when a specific antigen is present into the helper T cells. So the helper T cells recognize the antigen. That is the first step: recognition of the antigen by the helper T cells, by a specific protein receptor cells, that TCR cells. And after the recognition of the receptor cells, the changes or activation of the T lymphocytes occurs. Activation of the cell, proliferation of the T cells occurs, and that which enter into the circulation. So at the same time, macrophages produces the interleukin, and that which also stimulate or proliferate the T lymphocytes, and that which enter into the circulation and helps for the proliferation of other T cells like cytotoxic T cells. So first activation of the helper T cells, and then proliferation, and then which enter into the circulation, and that which stimulate the other helper T cells like cytotoxic T cells. And cytotoxic T cells that which kill directly by engulfing that microorganism by producing this lysosomal enzymes that which directly kill the microorganisms. That is the function of cytotoxic T cells. Sometimes these cytotoxic T cells that is specific to the viruses, sometimes the viruses are entrapped in between our normal cells, so it will kill the normal cells also. So at that time, the regulatory T cells function, that is suppressor T cells, that which suppress the action of killer T cells. So these are the functions. And memory T cells are the T cells that sometimes when antigen is that which stimulate the T cells and that which remain in the lymphoid organs, lymphoid cells, and the next attack of the same organism that which come the body the next time, and then it directly that which stimulate or proliferate the actions of T cells, memory T cells. And the second time it is more easily or more stronger than the first time by the help of this memory T cells. So this is the way that which the cellular immunity works. So helper T cells, cytotoxic T cells, then regulatory or suppressor T cells, and the killer last one memory T cells. That is the function of this cellular immunity. So that this is the mechanism of cellular immunity. And next slide. So the mechanism of humoral immunity. So the question may ask like immunity, types of immunity, and explain in detail about cellular immunity, or explain in detail about humoral immunity. That like the question may ask. So if the question is like a mechanism of humoral immunity, then you have to answer this question. You have to answer this and this one, humoral immunity. So humoral immunity means here the presence of antibodies are involving in the humoral immunity. B lymphocytes are involving. So the B lymphocytes are two T lymphocytes in the cellular immunity, humoral immunity. B lymphocytes are involving, and these B lymphocytes are that which produce the immunoglobulins or gamma globulins. സൗണ്ട് കഴിഞ്ഞില്ലല്ലോ. ഇല്ല, ഇപ്പൊ കഴിയും. So this humoral immunity, why it is known as humoral immunity? Humoral immunity because these antibodies are transported through the lymph and the blood, and in Latin this lymph and blood are known as humors. So this working through the humors, so it is known as humoral immunity, and these immunoglobulins are produced by the B lymphocytes. There are when an antigen come into the body, then the specific antigen that which stimulate the helper T cells, and the B cells also activation occurs. The B cell recognizes the antigen and the stimulation and proliferation of B cells, and the B lymphocytes that which proliferates and into plasma cells and memory cells. Plasma cells and memory cells, plasma cells that which produce the immunoglobulin or antibodies. So the humoral immunity works through the antibodies, and the main antibodies are IgG, IgA, IgM, IgD, IgE, different types of immunoglobulins, Ig. So this works through these antibodies. So this here humoral immunity works through these two mechanisms, mainly direct attack and the complement system. These are the two mechanisms, direct attack and the complement system. So through the direct attack, different actions are there. So direct attack, these antibodies directly attacking the invading microorganism by the functions like processes like agglutination. Agglutination means that which from the when an antibody that which form when RBC or any foreign body that which form clump by the antibodies that form the agglutination and that which block the attack of the microorganism, agglutination, and held together by a clump, which already we know that agglutination that which occur in the blood grouping that we know. So agglutination and then precipitation. In precipitation, these antibodies that which convert the this tetanus toxoid is a soluble form and it converted into insoluble precipitate form, that is precipitation. And then is the neutralization. In the attacking microorganism is there, and here the toxic sites are neutralized by these antibodies. So neutralization is there, and then lysis. Lysis is the most toxic form, and in this lysis, that which breaks or rupture the cell wall of the microbes. So direct attack is the first one. That's first direct attack of the antibodies. And next one is the complement system. Complement system is having three mechanisms: classical pathway, lectin pathway, and alternative pathway. So classical pathway, in this different types of enzymes, classical enzymes are involving, and it is in the cascade of mechanism you already told. So here different types of enzymes, plasma enzymes are involving, like C1 to C9. And here binding of C1 with the antibody that which stimulate other enzymes, C1, C2, C3, like plasma enzymes are there. And this plasma enzymes activation and that which produce opsonization, lysis, and chemotaxis, agglutination, and neutralization. You already told different mechanism. And this classical pathway, this complement system or complement method is more stronger than the direct attack. So this complement system is more stronger than direct attack. And then next one is the lectin pathway. Here the mannose binding lectin combines with the mannose or fructose in the wall of the invading organism, and that which kills the organism. And alternate pathway, here the binding of factor 1 with the polysaccharide that which activate the enzyme C3 and C5. So this that which kills the invading organism. So these are the mechanism of humoral immunity: direct attack and the complement system. And complement system is more stronger than the direct attack. And the complement system is again classical pathway, lectin pathway, and alternate pathway. This is regarding the humoral immunity. Next slide. So these are the important area we should cover before going for the exam. And other areas, bone marrow types and functions of bone marrow. We already know in the anatomy we will study the bone marrow types and functions. And blood groups, it's also important. And different types of blood groups. Karl Steiner's law, Land-Steiner's law is there, and platelets and its functions, anticoagulants, and hypersensitivity. This area you should cover before going for the exam. These are the important areas you should cover. And I think for an exam purpose, it's enough. This is an important area. RBC, functions of RBC, I didn't mention all about, and granulocytes, agranulocytes, different areas are there, but most probably from these areas, the questions we can see. In the previous years, we saw the questions from this area. Anticoagulants may ask, hypersensitivity, you cover, that is allergic response, hypersensitivity, different mechanisms are there. Blood groups, erythroblastosis fetalis. Just read that portion before going to the exam. I think the most important area covered for the exam. And so I think it's the last slide. Next slide. Okay, I think it's the last slide. So prepare for the exam. The Panchajanya that which blows at the time before the war. So this Panchajanya, the online platform, is conducting this type of online lecture series that before the exam, because it's a war. Exam is just like a war. So this Panchajanya that blows at the before the exam. So all the best for the coming exam. All the best prepare well and study well, and all the best. Thank you.
Thank you, sir. Thank you so much for the session. Now we are coming to the end of the session. Sir, we are very glad to have you in this session today. On behalf of our Panchajanya organization, we are very thankful to you for accepting our invitation to conduct this session today, sir. This session will be definitely useful for the students who are preparing for the upcoming exams. Sir, you have explained the concepts in very easy way. Once again, thank you so much, sir. We'll wind up the session now. Once again, thank you so much, sir.
Thank you. Thank you all.
We'll wind up the session, sir.
Okay.